The heart is the vital organ par excellence. We can feel it beat throughout each day of our lives. Although the heart rate adapts primarily according to our physical activity, it also fluctuates under the effects of psychological factors, such as stress. The heart serves to pump blood around our body, but before this discovery was made, our ancestors credited it with other functions, the most notable being that it contained the human soul. In 2005, INRIA researchers from various fields, automatic control, mechanical modeling, scientific computing, and image processing, launched CardioSense 3D, an ambitious research initiative in collaboration with clinical practitioners and industrialists. This research project is focused on constructing mathematical and numerical models of the heart, and then personalizing these models using clinical measurements specific to each patient. As cardiovascular diseases represent the leading cause of death in industrialized nations, the objective is to facilitate diagnosis and to better anticipate the effects of different therapeutic strategies. The heart is host to complex electrical and mechanical activity. It is in fact a muscular pump that is controlled by electric signals. Its function consists of pumping part of the blood volume into the vascular circuit with each heartbeat. A cardiac cycle has 4 principal phases: ventricular filling, isovolumetric contraction, ejection, and relaxation. Electrical activation is triggered at the sinoatrial node, a natural pacemaker located at the top of the heart's right atrium. To begin with, the electric signal spreads into the atriums. It then reaches the lower part of the ventricles through a rapid conduction network, the bundle of His leading to the Purkinje fibers, and then propagates in the cardiac cells at a more gradual pace, traveling thus through all muscular tissue in just a few tenths of a second. This excitation causes the muscular fibers that form the cardiac muscle structure to contract. More precisely, when we look into individual cells, or cardiomyocytes, the contractile function is performed by myofibrils, whose elementary entity is known as the sarcomere. To evaluate the condition of the cardiovascular system, examinations and exploration methods of varying complexities can be envisaged. Pulse and blood pressure measurements are routine clinical examinations that provide useful but limited information. Other exploration methods, with varying degrees of invasiveness, can be used to observe the electrical and mechanical phenomena in greater detail. Electrocardiograms, or ECGs, provide an external measurement of the heart's electrical activity. Electrodes placed at different points on the body record the electrical potential variations on the surface of the skin. The ECG is composed of the trace of these potential variations, that are in fact multiple, since several leads are employed. 3 principal events can be observed in an ECG curve. The first, known as the P wave, corresponds to the activation of the 2 atriums. The second is the QRS complex and reflects ventricular activation. The third and final T wave represents ventricular cell repolarization. The electrical system then returns to its initial state, and in turn the cardiac muscle relaxes. Different methods can be employed to take dynamic measurements of the heart's mechanical activity. Scans using ultrasound sent by a probe provide moving images of the heart. In particular, 3D ultrasound scans are used to obtain precise ventricular volume measurements while the heart is beating, and to measure certain cardiac tissue deformations. To obtain even more detailed and precise information from the perspective of planning medical interventions, research is being conducted at Guy's Hospital in London with collaboration from INRIA and Philips.
Our research is trying to bring in additional information to the electrical information in terms of the shape of the heart and scar and how it contracts a new technique that we've developed called XMR, where we combine both MR imaging and X-ray imaging together, allows us not only to get the electrical information from inside the heart from the catheter, but also the anatomy, the shape of the heart from the MRI, and combine the two.
The XMR system has several advantages compared to a traditional electrophysiology catheter laboratory. My research here at Guy's has focused on using the XMR system for image fusion to combine anatomical information from MRI and live X-ray images.
This complex data has great potential in helping us treat these patients better. And through our collaboration with the research scientists in India, we're now able to look at this data in a much more informative way.
The description level mainly targeted is macroscopic, that is, the tissue and the organ as a whole. Nevertheless, to understand and model the relevant physical and physiological phenomena, it is, in our view, crucial to initiate the approach at the very source of these phenomena, that is, at the cellular or even molecular scale. This form of modeling is qualified as multiscale. Let's begin by seeing how it can be applied to electrical activity. The cellular membrane constitutes a barrier for electrically charged ions between the interior and exterior of the cell. Nevertheless, certain chemical species such as sodium Na+, potassium K+, and calcium Ca2+ ions can cross this barrier using specific ionic channels. This means that devices comparable to pumps and exchangers actively induce these passages, and thus cause a difference between intra- and extracellular potentials, known as transmembrane potential. This potential, which spreads from one cell to another, forms a wave that is sustained through activating ionic channels. This is known as action potential. These phenomena are generally modeled using an adapted electrical circuit type scheme. In this scheme, the Cm element represents the electrical capacity of the membrane as an ion barrier. The other parallel channels correspond to the active elements, gates, and electrical sources that are used for the ion's passage. At the tissue scale, cardiac cells form a network surrounded by extracellular fluid. From a modeling point of view, the intra- and extracellular media, omega I and omega E, separated by the sigma membrane, can be distinguished in tissues. All are represented in the form of a very small structure that is repeated periodically. The intra- and extracellular potentials phi I and phi E are governed by Laplace equations with boundary conditions that incorporate the membrane's electrical properties. Through a process known as homogenization, we can obtain an electrical description of the tissue in terms of its mean intra- and extracellular potentials. These quantities will satisfy partial differential equations known as bi-domain equations. We can ultimately obtain a macroscopic description of the organ with all its different compartments. This knowledge is formalized by a mathematical model that schematically describes the organ's conformation. It is in this geometric domain that the bi-domain equations are posed. To understand and model the behavior of mechanical cardiac tissue contraction, we resume our analysis at the smallest relevant scale. In sarcomeres, the proteins responsible for contraction are organized in filaments of two types: thick filaments, most of which are made up of myosin, and thin filaments, principally composed of actin. The thick filaments are composed of multiple protuberances, or heads, that are liable to create chemical bonds at specific locations of the thin filaments under the influence of electrical activation. These chemical bonds, or actin-myosin bridges, and their associated forces can be modeled individually. We can now return our attention to the sarcomere by aggregating the behavior of all the bridges, which causes the thick and thin filaments to slide along one another, and in turn causes muscular contraction or relaxation depending on the direction. This collective behavior is modeled by Huxley equations, where a judicious choice of the bridge's creation and destruction frequencies, F and G, leads to a dynamic relation between elongation, force, and electrical activation. But other constituents from the cardiac tissue and cells must also be taken into account to correctly represent the average or homogenized behavior of tissue at a local level. This can be represented by a mechanical rheological scheme that integrates the various contributions in an assembly of elementary mechanical entities, like springs connected in series or in parallel. The tissue is now the intermediate organization level between the cells and the organ. Its conformation plays an important role in overall behavior, particularly regarding the specific layout of muscular fibers. Fundamental mechanical equations are used to describe the manner in which the stresses and forces are in perpetual dynamic equilibrium in the muscular tissue. We thus obtain, by integrating the contractile behavior model previously described, equations that govern the motion of cardiac tissue through space and time. As with the electrical model, a complete description of the mechanical model necessitates good anatomical knowledge of the organ, with its different compartments and the valves that separate them. Here again, it is the geometrical model that defines the domain in which the mechanical equations are considered. Once all the modeling elements required to describe the targeted physical and physiological phenomena have been formulated, we can carry out numerical simulations of the heart's electromechanical behavior. To this end, we complete a model discretization step, during which, in essence, we summarize the unknown quantities, such as displacement and electric potential, using their values in a collection of vertices in a subdivision of geometry known as the mesh. This step must be conducted with great care, as it is crucial in order to obtain accurate approximations and efficient computation methods for the simulations. Of course, to successfully use the model in numerical simulation, the directions of the fibers in the mesh must be identified, since they intervene as previously described in equations stemming from physics. We can now carry out a complete electromechanical simulation throughout the course of a heartbeat. Only ventricles are taken into account in the simulation displayed here. The electrical activation, shown in color, is initiated, just as it is in reality, in an area located near the apex and at the endocardium that represents the endings of the Purkinje network. The simulation results also provide performance indicators of the cardiac pump, such as how ventricular pressures and volumes evolve during the cycle. We can also obtain other quantities that are useful for a more detailed analysis of physical phenomena, in this case stresses in the tissue. The model can also represent pathological scenarios. Here we can see the effect of what is referred to as a left bundle branch block. This conduction problem in the left His bundle branch, generally caused by an infarct, implies that the electrical activation, shown here in color, is only initiated in the right ventricle, which leads to a late contraction in the left ventricle. This desynchronization, which impairs the efficiency of the cardiac pump, can be reduced by implanting a pacemaker that stimulates with each heartbeat the area where the electrical activation is deficient, thus resynchronizing the function. So that the numerical model of the heart can be used for enhanced detection and treatment of pathologies, it is necessary to adjust it using the available measurements of any given patient. This personalization of the model particularly concerns the morphology of the organ and the different model parameters that condition its behavior. It is therefore no longer sufficient to simply carry out direct simulations of a generic model. Instead, this model must be adapted so that the simulations correspond as closely as possible to the clinical measurements. This type of so-called inverse problem is at the crux of the preoccupations in the CardioSense 3D initiative. The first step consists in adjusting the geometry of the model to fit the patient's anatomy. For this, we use segmentation techniques on medical images that distort the mesh globally, then locally, to mold the shape of the patient's heart. This process is often difficult due to the poor contrast and resolution of the medical images. This means that robust methods must be developed. To successfully adjust simulations to the patient, in addition to anatomical data, functional data must also be available, particularly regarding motion details while the heart is beating. Tagged magnetic resonance imaging provides valuable detailed data concerning this point, with images in which a series of grids are drawn on the tissue, making it possible to monitor its deformation in a precise manner. Using the XMR system described earlier, we can obtain functional data, not only for cardiac motion, but also for electrical activation. Here we can see an MRI scan showing tissue tagging and electrical activation maps produced using endocardium measurements of the left ventricle for a patient suffering from a left bundle branch block. We can now make direct comparisons between these measurements and the results produced by the personalized model that has been manually parametrized to represent a trigger delay in the activation of the left ventricle. In some ways, data assimilation represents the culmination of this methodological chain by automating coupling between the model and data. Here is a preliminary example in which we use the model described earlier to simulate an infarct represented by reduced contractility parameters in two adjacent areas of the cardiac tissue. We thus obtain altered functioning, seen here through the contraction stress in the tissue during a heartbeat. This reference simulation is then used to generate data, in this case displacements, associated with some measurement points, data that we would be able to draw from clinical measurements like tagged MRI. Using these so-called synthetic measurements, the data assimilation problem consists of identifying the contractility values used in the reference simulation. Here we resume by setting a homogeneous nominal contractility value. The optimization algorithm used adjusts the value of the parameters by making successive comparisons between model simulations and measurements. Of course, this requires further work, since ultimately we must be able to use real measurements, but these results are already promising.
In collaboration with INRIA researchers, we are conducting studies to compare simulation outputs with clinical measurements. For us, this work is extremely important and highly interesting. The simulation results are very well matched with the initial physiological data we are gathering from observations in cardiology. We are also working with INRIA to perform an even more precise model validation, primarily through experimental campaigns. We think that the potential outcome of this collaboration is very important. Once our model validation has reached a satisfactory level, information supplied to doctors will be far more complete and more detailed than information available in the data. Doctors will also benefit from the model's predictive capabilities, and as a result, they will be able to predict the development of the cardiac function in a patient over the course of time with far greater precision and reliability. They will also be able to predict the effects of therapy or surgery. This is why I truly believe that the scope is enormous, both in terms of diagnosis and prognosis, but also to successfully set up an appropriate therapy strategy for our patients.
This research, launched a few years ago as part of the CardioSense 3D initiative, is already producing tangible results, even though it is clearly a long-term project. In particular, in modeling terms, the diversity and complexity of the physiological phenomena represent a source of constantly renewed challenges. Among all the conceivable developments, our priority is to take the intra- and extracardiac bloodstream into account and to model perfusion and metabolism as well as damaged tissue's evolution through time. These objectives, together with the project's future developments, go hand in hand with progress in imaging processes and medical practice, since modeling, measurements, and clinical applications are indissolubly connected in our approach.